It's a pleasure to be here today. Welcome everyone to this session of the NDF Speaker Series. My name is Noah Weisleder. I'm a professor and director of graduate studies in the Physiology and Cell Biology department at The Ohio State University, and also an investigator in our neuromuscular disease research center here at Ohio State. It's a pleasure to be here today and talk with you. Today I'm here in the capacity of a science advisor to the Neuromuscular Disease Foundation. I've been one of the people who has been advising the Neuromuscular Disease Foundation on scientific matters over the last couple of years. In addition, I also was headed up the granting process by the NDF for these last 2 years. And it's a good opportunity today to come and speak to the patient community and others about what is associated with the grant review process at the NDF, and some of the grants we funded in the past, some of the ones that we're funding this year, and some of the good that has done as we've been developing. The NDF has been contributing to developing different types of therapeutic approaches for the treatment of GNE myopathy. And I did want to point out, just in terms of this online session, we can take questions and answers. There should be a Q&A tab available. I have it right in front of me here. I will try to get to your question as soon as possible. There's no need to hold them to the end. If you need clarifying questions, I'm happy to fit them in as we go through. If I miss them, I will get back to them as quickly as I can. So please feel free to ask as many questions as you'd like. We'd love to have some exchange here with the patient community and talk about some of the great work that's taking place at the Neuromuscular Disease Foundation. So for today, I'm going to focus on some of the major milestones that have taken place in research in GNE and GNE myopathy over recent years, really going back almost 20 years now since the identification of the gene. So we're going to go through a little timeline of what's taken place there and how the NDF has contributed to that through some of its grant-making efforts to help to develop treatments for GNE myopathy, or HIBM. We'll go over some of the funded research projects that have taken place since 2020. Really, we had a bit of an uptick in the amount of grants that were funded in 2019 and moving forward. So sort of an inflection point there that I like to point out some of the projects that have been going on since then. In addition, talk a little bit about the grant review process that takes place in the NDF, with a specific example from this year's grant review process, and how the NDF goes about determining what grants to fund, and how it goes about making sure that is done in a robust nature that is conducted by world-class scientists. And then how we go about being transparent about presenting the results from that work and the grants that are being funded. And then finally, I want to spend a little time talking about how those grants help support the International Gene Therapy Development Program, which is a major initiative by the NDF to produce a gene therapy for the treatment of GNE myopathy. So I just want to do a couple introductions here. As I mentioned earlier, I'm one of the science advisors for the Neuromuscular Disease Foundation, but there's two other people who are really important for the efforts that take place through the NDF, particularly as it relates to the gene therapy development program. That's Angela Leck and Rich Horgan. You can see them here. They both have day jobs. Angela Leck is the vice president of research for the Muscular Dystrophy Association, and there she heads up several aspects of their work on a wide variety of different neuromuscular diseases. Richard Horgan is the founder of Cure Rare Disease, which is another nonprofit that's associated. That its mission is to develop different types of therapeutics for the treatment of rare neuromuscular and other types of rare diseases. So he has a lot of experience with product development and moving things towards the clinic. So both of their expertise has been very valuable to the NDF as they've been developing the sponsored gene therapy to move towards the clinics for the treatment of GNE myopathy. And I'll talk a little bit about the end here, but principally what we'll talk today is generally about the grant making process. But I would like to tell you how that ties into the work that Angela and Rich have been doing for this gene therapy development program. So one of the things I like to, sorry about that. One of the things I like to go over is some of the major milestones in the GNE research community. So, we look back, over 20 years now, before there was the first cloning of the GNE gene that was done by Stella Mitrani Rosenbaum at Hadassah University. That was a major step forward. It opened up the possibility to treat this disease in a lot of different ways. And Stella's been a very important part of the NDF and their efforts for many years now. And since that time, since the initially characterization of the disease itself, there's been a lot of steps forward, as you can see here from this timeline. The approaches done by the Nishino laboratory in Japan, looking at ways to use supplementation therapies for the treatment of the disease using mouse models. The generation of first generation gene therapy constructs, several different types of supplementation theory experiments and clinical trials. So one of which, as you're probably familiar with, the sialic acid extended release trial by Ultragenyx. Many of these benefited greatly from the work of the NDF. As you can see, those findings that are boxed off here in blue, the larger blue shading, that's during the NDF's funding time. And you can see how over that time, and increasingly, the number of major milestones in the GNE research field have benefited from funding that's come through the NDF. So really, when you look back at the work that's been done in this field, it's really been accelerated greatly by the work done by the NDF. So that's really been one of the reasons that we're starting to make progress in the treatment of the disease, not just through potential supplementation, but also in the rapid progress being made in developing gene therapies to treat the disease. So, as I mentioned, the principal role that the NDF does in facilitating the development of therapies for GNE myopathy is through making grants. And that's really been an important element of what's brought more investigators in the field and kept investigators working this area so they can have active research programs that can move towards treatments for the disease. So that's not the only thing the NDF does. The NDF also plays a very active role in bringing together these investigators, convening them with different types of scientific meetings, different types of online meetings to facilitate interactions between the investigators to avoid duplication of efforts and to really hopefully accelerate the path towards different types of treatments. In addition, they've established several resources that are available to the broader scientific community that are interested in working in GNA myopathy. And we'll talk a little bit about those as we go through the individual grants, but that's also been a very useful thing. And my laboratory also does GNE research, and it's principally because of these resources that the NDF developed, which made it very easy for my lab, which had been interested in working in Duchenne muscular dystrophy, to begin to work in GNE myopathy. So if we look at the grants that have been provided, as I mentioned, there's a long history now. The first grant from the NDF went out in 2007. And there's been funding that's been run continuously over that time through the NDF to help support different types of approaches. That really has been key, as I mentioned, bringing new investigators into the field. That's been one of the real initiatives, particularly over the last couple of years, is trying to make sure that we support and bring in more investigators into the GNE myopathy field. In order to have an effective development of treatments, it's really important that you have a robust scientific community in order to both develop and then test those therapeutics. The 2023 to 2024 funding cycle is underway. I'll talk more about this later, but in that we plan to give out individual awards of up to $75,000 per award. We were looking potentially to do larger projects. There wasn't sufficient funding to do that this year, but hopefully in the future we can continue to give out larger projects as well. And we were able to fund 4 grants out of the 8 projects that were potentially available for funding. I've broken out here a few of the projects that have been funded since 2020 by the NDF. I think this is useful because it gives you an idea about the importance of these different projects as it relates to developing new treatments for GNE myopathy. Now, as I mentioned earlier, there was really an inflection point in 2019 when the research efforts of the NDF, I would say, moved to another level. The funding levels went up. and more investigators were brought into the field this way. And that was really the foundation by which we began to build the effort that became the gene therapy development program. By laying the foundation with several important things that were necessary to develop for the GNE myopathy gene therapy, but also not just forming the foundation, but also providing the actual experiments that are necessary to move that development of gene therapy forward. So I just point out a few specific ones here that are very useful for that. A couple ones that I mentioned earlier, Dr. Mitrani Rosenbaum. Her work has focused over these last few years in developing mouse models of GNE myopathy. It's very important to have animal models of a disease in order to be able to test if therapies work before you move into humans. That's one of the requirements that the Food and Drug Administration of the United States has in order to be able to move a therapy forward, is showing that it works. And usually that's done in mouse models before you move into, or other animal models before you move into human patients. So one of the challenges of the field has been that there is not a mouse model of this disease that shows all of the muscle pathology that you would normally see in a patient with the disease. So that's one of the efforts that Stella's been conducting in her laboratory, developing that mouse model, a new type of mouse model that will we expect to show a muscle phenotype, that is muscle pathology, a disease within the muscle, that you're able to use to test whether therapies work or have a reasonable expectation of work in human patients. Dr. Paul Martin's been doing a similar approach as well. He's at Nationwide Children's Hospital, which is one of the preeminent institutions in the world for development of gene therapies against neuromuscular diseases. And he has been developing another mouse model, similar one, with the intent of also finding a mouse model that could be used for developing both gene therapies and other treatments for GNE myopathy. And you can tell from the fact that a lot of resources from the NDF have gone towards this. This is a really vital effort. So this has been going on for a few years now, developing these new mouse models to be able to test different types of therapeutics. In addition, there's been a lot of development of different types of biomarker approaches. Biomarkers are ways that you measure if a therapeutic is working in an animal model or in patients during a clinical trial. So these are really vital to understand what you need to have to be able to test whether a therapeutic is working or not. So those have taken place in a number of different laboratories. Dr. Kelly Crow's laboratory at Xavier University, Dr. Horst in Germany, as well as Dr. Monko Leck at Yale. They've all been working on developing new type of biomarker approaches to measure the effects. In addition, we have been developing the gene therapies as well. That work's been going on both in Dr. Julie Kudel's lab at the University of Washington, as well as some work in Dr. Monko Leck's lab, again at Yale University, and also looking at some of the work that's been taking place at the National Institutes of Health that also has involved both developing biomarkers and helping to measure the changes that take place in some of these gene therapy testing samples. That also involving, of course, measuring different biomarkers. So so there's been a lot of work going on here that's provided a really stable platform in order to begin to build not just gene therapy development programs, but any type of therapy program. And ultimately, we'd like to develop a lot of different types of therapies, because the more type of therapeutic approaches you have available, the more you have being developed, the more likely you are to be able to come up with one that is going to be able to treat patients in the future. I see that there's a question in the question and answer section. That question is, can AI help us get away from animal models? That's a really good question. So I'm sure you've all heard a lot in the news about AI and how that's thought to be a way to revolutionize a lot of different aspects of what takes place in almost every field of human endeavor, right? But certainly in science. And there's been a lot of advances made using different types of artificial intelligence approaches. One of the challenges with that is it's very difficult to model what takes place in an animal using computer simulations. And that's something we've tried to do a lot of as a field for many years, really. How AI will be able to be helpful in that is not entirely clear yet. It's at this point, at the stages of development, it's difficult to see how AI would be able to model what takes place in a full organism. Computer modeling has sometimes it's a challenge to model just what one protein does, and we're made up of a lot of proteins and a lot of other types of macromolecules. So AI is certainly has promise there, but probably is very much in the future before we be able to not use animal testing completely to ensure not only that a therapy is effective, but also that it's safe. I suspect animal trials will exist for quite a while into the future. Hopefully, we can use less of them using AI, but I'm sure we'll still have to have some animal trials into the future. One new technology that actually does have a lot of promise for reducing and potentially eliminating the need for animal trials are different types of what we call biomimetic organs or biomimetic cultures, or it's sometimes called tissue on a chip or organ on a chip. That is technology where we use stem cells, adult differentiated stem cells from donors that are through various approaches made into a different type of biomimetic tissue. That's relatively easy to do for skeletal muscle. It's harder to do for some other tissues like the brain. But those sort of biotechnology approaches can be used to both screen for new drugs and to test the toxicity of other drugs. And that sort of technology is already in use in different laboratories and pharmaceutical companies around the world. So that's certainly not only helping to reduce the number of animal models that are used from the point of view of reducing the use of animals, but also to provide a more translational approach, because as maybe some of you know, sometimes drugs behave differently in animals than they do in humans, and that leads to frequently you'll see that a drug in a clinical trial won't show the same effects that you see in the animal studies that facilitated that clinical trial. So one of the hopes of these sort of biomimetic systems that use human cells is that you can reduce the number of times that you'll have a drug that doesn't work on humans. That technology is relatively new, so it's hard to say for sure at this point, but there's certainly some reason to believe that these sort of approaches will reduce the number of animal trials in the future. And there's investigators working on that for GNE myopathy. There's a group in Korea, another group in Indiana University School of Medicine, that's been involved in developing iPSCs, adult stem cells from donors, that can be used for these sort of approaches. Also, the gene bank, the gene bank and the myoblast bank that the NDF has developed and is housed in the Leck lab at Yale. Those cells can all be used for these sort of biomimetic tissue approaches. So that's certainly something that maybe in the future we could reduce the number of animal studies necessary. Hope that answers your question. If there's any other questions, please feel free to put them in the Q&A box. So beyond those projects that have been funded in recent years by the NDF, there's also a robust program going forward to continue funding to help support the development of therapeutics for GNE myopathy. So this comes to the current day here. So with our granting process, it's changed a little over the years, as you might imagine, as we've tried to refine it. But the way that we have done the NDF has done these sort of analysis over several years is to request applications. They put up a request for applications on the NDF website and also advertise those on their various mailing lists sent out to both to the patient community and the scientific community in order to solicit grant applications. As I mentioned earlier, this year we were looking for grants of up to $75,000 per laboratory for this granting year. We had wanted to do larger projects as well. However, the funds that were available could only fund $75,000 per laboratory. So one thing we added this year was a letter of intent screening. So that was intended to reduce the burden on investigators spending time writing applications. if it wasn't corresponding to what the NDF was looking to fund. So we received 10 different letters of interest or letters of application, letters of intent from the scientific community. Those letters were screened by the Scientific Advisory Committee at the NDF. 8 of those letters of intent were asked to apply a full application, and those were looked for by review by the Scientific Advisory Committee. In each case, there 3 to 5 of the Scientific Advisory Committee members provided a score about how well or how much that would impact the development of therapeutics for the treatment of GNE myopathy. And after those were scored, the top 4 of those grants were able to be funded. So almost 50% of the final applicants were selected for funding from that came in. Just to sort of show what that's like in relation to the funding that might occur in other organizations. When you look at the National Institutes of Health, they only tend to fund around 15% of the applications, depending on the topic that come into their, in many cases, less than 15% of those that come in. So at least there was enough funding to support a high percentage of the applications that came in this past year. Wanted to point out here too is that these were reviewed by the Scientific Advisory Committee. The members of that committee are listed here. I won't go through all of their credentials today because that would take too long. They all have so many credentials. But if you're interested in reviewing those, that information is all on the NDF website. The link directly to it is there, but it's very easy to find under the leadership tab. But I wanted to emphasize the quality of the Scientific Advisory Committee that's available to the NDF. It's a real asset to the NDF. As I mentioned earlier, Dr. Mitrani Rosenbaum is on there, obviously a leader in the field who originally isolated the gene responsible for GNE myopathy. Did a lot of that work with Dr. Argov. He's also on the committee as well. And as I mentioned earlier, several leaders in the field, as well as both scientifically and in the medical field, Dr. Perry Shea, Dr. Mozafar, they're all clinicians as well who treat GNE patients, GNEM patients every day. So it's good to have not only the world-class scientific expertise, but also the world-class expertise as it relates to the clinical aspects of the disease too, available to the Scientific Advisory Committee. This is a real asset to the NDF, and we appreciate their efforts. So when we went through that process, as I mentioned, each one is assigned a score based on its potential impact, and we funded those that were the top, that scored the highest on that scoring of the impact on the treatment for the treatment of GNE myopathy. So there are 4 different grants listed here. That's if you can see that here, we funded 2 grants at the maximum amount, 2 that were less than that full amount that didn't require the full available amount of funds in order to accomplish their projects. So if we look at that, I'm going to go through each one of these individually, but you can see that there's projects that are focused both on developing new gene therapies, but also on developing those mouse models that I talked about earlier, which are really vital for the testing of not just of gene therapies, but also those that are involved in the any other therapy for treating GNE myopathy. So the first one that I want to talk about is about codon optimization and producing a better GNE gene therapy approach. So this work was done by Julie Cradell. She's a rising star at the University of Washington in the Department of Neurology. She did a lot of her postdoctoral work. She did that in Jeff Chamberlain's laboratory at the University of Washington, which is certainly definitely one of the leaders in gene therapy as relates to a lot of different types of neuromuscular diseases. So she had excellent training. She's an excellent investigator. And coming into this, being able to have her come into the GNE field through the efforts by the NDF, I think has been a real credit to what's taking place through the scientific effort by the NDF. So her work focuses on coming up with next generation gene therapy approaches. I mentioned earlier that there have been efforts with gene therapy before, but the technology has advanced quite a bit over the years since that initial effort. And what Julie brings to these efforts is really showing that these state-of-the-art efforts can improve the quality and the efficacy of the gene therapy approaches that are being developed. So she's taking several different approaches. She's looking at different types of optimization of gene therapy that can cause these gene therapies to work better. One of those is called codon optimization. That's how you change the sequence of what you'll be using in the gene therapy vector. That allows you to have even more robust expression of that gene therapy. Also, she's looking at removing different types of sequences that can lead to immune responses from gene therapies. And that's really vital too. We want to avoid as much as possible, avoid immune responses, because that's really one of the ways that you can have toxic effects from gene therapies. So these approaches that she's conducting in her laboratory are going to produce a much more effective gene therapy approach and be able to guide some of the work that is being done by the International Gene Therapy Development Program. And also, she's going to produce her own types of gene therapy approaches that might provide another opportunity to develop gene therapies that could be used in patients in the future. I mentioned earlier, Paul Martin at Nationwide Children's Hospital is also working on a mouse model of GNE myopathy. He's trying to address this challenge where there are several mouse models that have been developed for GNE myopathy, but unfortunately those don't show the same sort of pathology in the muscle that we see in human patients. So this has always been a question in the field, but it's always been, and trying to develop a mouse model that actually shows that muscle defect has been a really important part of what a lot of laboratories are doing. And certainly the NDF is at the vanguard of that by funding both Paul Martin's work as well as Stella Mitrani-Rosenbaum's work. So that's what's taking place in his laboratory over this year. He's making a inducible knockout model. That means he can remove the GNE gene function at different stages of the mouse's aging. And he hopes that this model will help allow for not only a mouse model that would develop muscle pathology, but also one that can be used for testing gene therapies and other types of therapies that he's interested in testing in his laboratory. So that's work that is taking place in Paul Martin's academic laboratory that's sponsored by the NDF over this year. In addition, similar work, but a different mouse model is being developed in Stella's laboratory at Hadassah University. As I mentioned earlier, it frequently is much more efficient and actually more likely to be successful when you take multiple shots on goal, right? You need to be able to have in order, if one of the things that's limiting your development of therapeutics is the lack of this mouse model, the NDF has taken the approach of moving towards having multiple mouse models available. There's slight differences in how this is being developed, so those differences may make a big difference in the ultimate outcome. So Dr. Mitrani-Rosenbaum, she's developing a mouse model as well that will hopefully have GNE defect, or that the removal of GNE function from these mice will lead to defects in skeletal muscle, and that will allow, again, a new model that we can use to test different types of therapies for GNE myopathy. Monko Lex Lab, as I mentioned earlier, has really been a workhorse in several different aspects of work done by the Neuromuscular Disease Foundation over the last few years. That includes developing both the gene bank and the cell bank that helps other investigators get the resources they need to conduct their studies, but also in doing some foundational work, looking at whether CRISPR gene therapy might be efficiently deployed against GNE myopathy, and also developing some new biomarker assays as well. And the work in his laboratory over the last couple of years has really been focused on supporting the International Gene Therapy Development Program. So one of the ways that we try to minimize the costs associated with the research done in developing a gene therapy is by working with collaborators, different types of academic laboratories that the NDF has worked with in the past, in order to conduct the assays needed on these gene therapy trials. So if you do these sort of studies with contract research organizations, companies that for-profit companies that work on this, they're much more expensive than if you work with academic laboratories. So that's one of the reasons that we have focused on working with academic laboratories to do a lot of these studies. And a lot of that work has been done in Monko-Leck's laboratory. And his laboratory there at Yale is doing some important pieces of work to help develop gene therapy this funding year. One of those is helping to determine what sort of a dose is needed for to produce gene expression in mouse models. And the other is continuing his work in developing potency assays in order to measure the effectiveness of these gene therapies. So these are two critical factors to help move along the gene therapy development program that the NDF is running. And we're fortunate that he continues to work in this area. Any questions on any of those grants at this point? Because I'd like to move into talking a little bit about the IGTDP, but if there's any questions now, I'd be happy to take them, or you can hold them to the end. I don't see any questions just now, but please continue to think about those. I'm happy to answer them when we get to the final bit of the presentation. But I did want to talk a little bit about the IGTDP. This gene therapy development program is a major initiative by the NDF. Its goal is to become a sponsor for the an investigational new drug application for AAV-mediated gene therapy replacement and be ready to conduct a clinical trial. The goal of this really is to generate data. That data would be necessary for producing a pre-IND package that would go to the Food and Drug Administration and be able to move towards developing the sort of things that are necessary to conduct a clinical trial for the treatment of gene myopathy. Now, this is not something necessarily the NDF can do by itself. Clinical trials in gene therapy are a very expensive operation. They would cost more than the NDF has funded since it began its funding in 2007. So this is a very difficult program that needs to be de-risked. The goal here is to develop a good understanding of the challenges that are associated with that, to get consent by the FDA to support the projects moving forward, and then be able to hopefully engage pharmaceutical companies or biotech companies to be able to help move this project forward, scale it up so it can have worldwide distribution. But it's necessary to have these studies to show that it's possible to do this study, and to be able to engage those companies and have them move this program internally there and help to develop this gene therapy so it can actually get to patients. Now, a lot of this work, as I mentioned earlier, has been coordinated by Dr. Angela Leck and Rich Horgan. That work has they put a great deal of work in facilitating this effort by their collaboration with both contract research organizations, and I mentioned earlier, different NDF-funded scientists that help to keep this program going forward. Now, I'm not going to go into all the details today. We'll have another session in the speaker series later this year in order to show that some of the progress has been made there. But I did want to talk a little bit about the overall arcing system that is being used. This is a program that's been going on since 2021, continues to develop here. The idea here is several different steps: the design of the gene therapy, the validation of that gene therapy through gene therapy testing, and then assessing the impact of that in different mouse models. That's what's taking place right now. Our hope is that the clinical development, the preclinical development, will have some data coming off that late 2023, 2024, which will allow moving towards developing a GNE gene therapy or a pre-IND on GNE gene therapy that can be submitted to the FDA to learn how much more work needs to be done in order to develop in order to get in order to determine the additional studies necessary to actually get the IND application in place. See a hand raised. Did you have a question, Tara?
Yeah, for the benefit of our patient community, can you elaborate more on the pre-IND package we need to bring to the FDA?
Yes, I can. Actually, I'll talk about that on the next slide. It's a good question.
Thank you.
Sure. I don't see any other questions there, so let's move on to the next slide to answer that question. So this is an update on the status of GNEM gene therapy. So this is exactly what we were just talking about earlier, what needs to be done in order to put together the pre-IND package. So that pre-IND package is the key elements here you see broken down into several different things that we need to have available for that. So several of these have already been accomplished. One of the key elements of that was selecting a mouse model and establishing a colony for that. This is done at Charles River Laboratories. That's a large contract research organization that works with different types of animal models. This is so the program that where the mouse model that's being used, that is what was called a knock-in mouse model. It adds in a mutation that mimics a human mutation. This is the M743T mutation. That mouse has been used in the past to validate some of the gene therapies that have been used, the first generation gene therapies that were done in earlier work. That work, since that work was engaged, and that project had been engaged with the FDA. At that time, the FDA felt that was a reasonable model of the disease. So that was the model that was selected while we're waiting to develop the additional mouse models that are being developed in Dr. Martin's and Dr. Stella Mitrani-Rosenbaum's laboratory. So first thing was to establish that mouse colony and select which mouse would be used. Then we had to identify the window of the GNE-related phenotypes. So we needed to understand what phenotypes in those mice we would be trying to rescue. And then we had to develop the gene therapy constructs to put into those mice. So that's the actual gene therapy itself. That's the virus that needs to be generated in order to produce that. So there's several steps to that. There's producing or designing what that vector is going to look like. There's producing the actual gene therapy material. That was done through another contract organization. That's Andolin, which is a major company involved in developing gene therapies or producing gene therapies. And once those vectors were available, they needed to be tested in that mouse model. And that's where this testing had gone. We did initial testing comparing these new vectors with those that were used in the original gene therapy approaches. And we were able to show that the new vectors were even more effective than the original vectors at producing GNE expression. So those were important steps forward, and those have been completed. What else needs to be done before we can do the pre-IND package is to complete the head-to-head comparison of this new vector with even a more advanced vector called AAV-MYO2. This gene therapy vector is improved for its ability to target muscle tissue. So that would require less dose, which would mean less cost to patients, as well as a better safety index for patients. So right now we're testing to see if that can be used. That's a newer vector. So we want to complete that before we go to the FDA, because that's something that they haven't seen as much of before. So we want to show that that is effective before going to the FDA with a pre-IND package. Once that's completed, then we'll do assays of that to confirm that it's working, that it's producing changes that would be expected in this mouse model. And that's what we see taking place here when we talk about hypocylation. That's one of the changes that takes place in mouse muscle and in human muscle, and to confirm that the gene therapy is functional. When that's completed, that allows us to prepare the pre-IND document, the package that will go to the FDA. Once that paperwork is completed, that can be submitted to the FDA, and then they'll have a meeting. That meeting with the FDA is where they'll tell the NDF and what other studies need to be done in order to be able to have an IND application to the FDA. So they'll after they get these results, they'll analyze that. They'll come back and say, well, we think that you need to do this additional toxicity study, or that you need to do this additional efficacy study. And once you know that, you can, those are the studies you need to complete in order to take your IND package to the FDA and ask them to be able to do a clinical trial. Does that answer your question? If there are any other questions about that? And if I go to the next slide, then this, I just want to give a quick update of the key takeaway messages here, and then I'd be happy to take any questions people think. So go ahead and get those questions ready. The key takeaway messages that I'd like you to take home with you today are that the NDF has supported some major milestones in its pursuit of the treatment of GNEM HIBM over several years since the initial funding in 2007, with a major increase in the funding that took place in 2019 and going forward. That has allowed the NDF to contribute to many of these important milestones as we move towards treatments for GNE myopathy. The NDF facilitates an open grant review process. It's driven by a world-class scientist and clinician review, and that is based on the most impactful science as judged by those scientists and clinicians. So the real focus there is that is going to most impact the development of therapies for GNE myopathy, and those most impactful studies are those that the NDF is funding. And then it's very clear, and then there's summaries of those posted on the website. We have them come and present at this speaker series, so we can maintain maximum transparency as it relates to the patient community. We really want to make sure that we update and let you know what's going on in these projects and how they're helping to move towards treatments for GNE myopathy. This year, the NDF is funded for additional grants that help support new gene therapy approaches, including the International Gene Therapy Development Program, which is a major initiative by the NDF. That program continues to move forward, and with the support of the grants from this year, we hope that in the next year we will be able to submit a pre-IND package to the FDA to begin moving forward towards the final IND package. That's what I'd like to go over today, and hopefully these takeaways are useful for you, and I'd be happy to take any additional questions.
Did you disclose when we will have our packet created?
Yeah, so the IND so it's difficult to set deadlines when it comes to science, right?
Yeah.
One of the things so I originally trained as an engineer, and one of the things that always when you're an engineer, you design systems. You build a system and you know everything about it and you can control everything about it. And we see a lot of this in a lot of human endeavor beyond science and engineering too, right? When you create a business or a lot of different types of things created by humans, we created them so that we have control over that. When you go to nature, when you go to biology, it can be much more of a challenge to understand things, because you're not really building things, you're discovering things, and you really don't know how discovery goes until you get to the final destination. So, and as I mentioned earlier, there's been some twists and turns in this process, even as we begin to better understand how gene therapies work.
Yeah.
So the one example of that is the moving from using an older vector to the newer vectors. And, that each time you do that, you have to take a few steps back in order to make sure that that vector is working. So those unexpected challenges always complicate timelines when it comes to developing anything really in the biological sciences. But right now, with the ongoing trials that we actually have, as Rich is fond of saying, in life, those studies going on with mice right now, assuming that they operate as expected, we do hope that we should have results coming in late 2023, the later part of this year and the early part of next year, 2024. And that those are the studies that ultimately we would need to move forward with the pre-IND package. So our expectation is that within the early part of next year, we'll be putting that package together and looking to get feedback from the FDA. The FDA has a pretty standard methodology for this. They require a few months to review a packet, but then they give you feedback in a timely fashion. So I hope that during next year, we'll be able to have a good idea what additional studies will be to move from the pre-IND package to the final IND package that would be able to move towards or be the permission to be able to do a clinical trial. Now, I need to emphasize again that there will be studies in between the pre-IND package and the IND package. Very rarely do you move directly to an IND package. The FDA always requires additional studies, but it's difficult to say what you need to do until they tell you. But there will be some time in between those as well. So that's a good question. But yeah, as you can see from this timeline, that we're looking to have some preclinical data in late 2023 to 2024. Other
Thank you.
questions? Yeah, you're welcome. I would also emphasize that there's some there's a real value in the questions from the patient community, and I'm more than happy to answer those at any time. My contact information is available on the NDF website. You can reach the whole scientific team by sending an email to the science email at the NDF as well. All of those are really valuable resources for us. We do like to hear from the patient community, and are happy to update you at any time if you have questions.
You're familiar with the National Institute of Health, correct?
That's correct.
Okay. Is there a reason why NDF decided to go through, are we ever going to touch base with the National Institute of Health at all?
Well, there's two main parts of the National Institutes of Health.
Yeah.
It's what they call the intramural and the extramural programs. So the intramural programs, the NDF has been engaged with them for several years. As I mentioned, we work with laboratories there in order to there's much great work that goes on there. One of the key elements to validate the International Gene Therapy Development Program, some of that work's taking place at laboratories at the NIH. And the NDF has funded that work taking place in those laboratories. So that's really been an important element. Another part of that is the MANAC trial that's going on with the partially developed at the NIH, but also through Leadian Biosciences. That has benefited from work done by the NDF in the past, but also the NDF has worked with them as well to help to have Leadian. I know there's been meetings before I was involved between scientists at the NDF and Leadian to help develop their clinical trials. So on the intramural side, the NIH proper, I guess, scientists who work at the NIH itself, there's quite a number of engagements between the NDF and the NIH. When we look at the NIH, the extramural funding, so the extramural funding are grants that they grant to laboratories mostly around the United States, but also around the world, to do research in a wide variety of different types of human diseases. And in that respect, we've been encouraging NDF investigators to pursue funding through that source. We've been advocating for more funding to go towards treatment of HIBM, GNEM. Obviously, there's a lot of different priorities at the NIH, so that's one of the things that's important to continue to advocate for, both having more NDF engaged investigators apply for NIH funds, but also to support them as they do that, and to encourage the NIH to also provide more funding for that. And that's one place where the patient community can get involved as well. The National Institutes of Health have several different mechanisms by which you can communicate with them as patients and as family members of patients. And they do listen to those, and they do pay attention to the amount of public input related to particular diseases. So one of the things that the patient community can do is to advocate for more funding from the NIH to go towards the developing treatments for GNE myopathy. So I'd encourage the patient community to do that as well.
Thank you.
Certainly. I see that there's another question. Let me see here. That's in the question and answer box. I guess it's more of a comment there. Thank you to everyone who put this together. I really appreciate all the efforts involved. Noah, thank you for your presentation. Remind me of Andrew Huberman. Well, thank you very much. I appreciate the comparison. Any other comments or questions? Happy to answer anything that comes up. And like I said, I mean, one of the things that I think is really important is to keep engagement between the patient community and the scientists and clinicians who are working on these sort of treatments. You know, I like to think that we try to be as approachable as we can be. We have these seminars. We put out white papers about the work that's going on. We have summaries of all the grants and their progress that go up on the NDF website. But like I said, we really do this for the patient community. And we want to make sure that the progress that's taking place at the NDF is communicated as well. And that you understand that we appreciate the insights you can give us into what it is to be a patient and the challenges that you face, and the challenges that are faced by the family members of patients as well. This is an important element of why many of us are in biomedical research, is looking at ways to find treatments for different disease states and and that's how we really help the patient community. So we appreciate any feedback that you have, and feel free to reach out anytime with that, and questions too. I mean, that's how one of the ways that we can keep engaged as well. Any other questions?
I don't see any raised hands or any questions in the chat box.
Okay, great. Well, I guess in that case, I will let people go. I do appreciate your time here. I know I can tell from the list here we have people from all around the world, some familiar names there as well. We appreciate you making the time and coming to the NDF speaker series. And as I mentioned, there are other ones coming up for the remainder of the year. We'll have one that's more focused on the International Gene Therapy Development Program, and also one from a new investigator in the GNE myopathy field. As I mentioned, we're one of the initiatives over recent years has been trying to bring more investigators into this field, and this is one of the ones from Indiana University that's interested in developing cell-based models for GNE myopathy, these are human induced pluripotent stem cells. So these are ways to be able to use human cells to develop therapeutics. As I mentioned earlier, that's one of the real advances in the overall field of biomedical research, has been the use of human stem cells for testing therapeutics. And hopefully that's something that will interest the community, and hopefully also something that will move these therapies along as we're trying to develop them. So I wish you all an excellent day. Thank you for your attention, and I look forward to talking with all of you again soon.